So we are starting this thing about bioinformatics. So what is bioinformatics? As the name indicates, it comprises of three different terms. First is bio, second is info, and third is matic. In case of biology, how many atoms we are dealing with? If I'm reframing the question, what do you mean by life sciences? What atoms or what molecules belongs to that particular bio or life sciences? And considering it, these five or six particular atoms are there. These are comprised of this biology thing. But if we are considering chemistry, whole periodic table is there, but we are not considering whole periodic table, except there are certain major atoms that are present in biology, specifically carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. These are the things which majorly form certain kind of biomolecules. In case of biology, we are considering six different atoms, namely carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. What is info or information technology? There is a separate branch. For example, biology is a separate branch, information technology is a separate branch, and mathematics is a separate branch. For example, so biology, we specifically focus on six different atoms. These are carbon, hydrogen, oxygen, sulfur, nitrogen. These are things. What about information technology? What we do about information technology, or what are the tasks performed in that particular field? So this is a database. So we are retrieving something. We are focusing on this thing, that is database thing, because our experiment is related to that. Means we can take out data from that particular database. At the same time, we can store also something. These two things are going simultaneously in case of information technology. And in case of mathematics, we are dealing with various algorithm preparation or the calculation thing. Along with that, there is X is there, that is statistics. So all the things combination is there in case of bioinformatics. So we are dealing with biology thing, we are dealing with computer science thing or the information technology thing, that is database creation, networking, all the things are there. Along with that, we are doing certain kind of calculation, or we are predicting certain, we are making certain kind of models that are based upon statistics in machine learning also. So all the things will comprise in this bioinformatics. Second question is, why we are doing bioinformatics? We are discussing three different things in case of bioinformatics. But first is what, second is why, and third one is how. So second question is why. Why we are doing bioinformatics? Because it's a part of syllabus or any other thing. So I'm first taking and second taking this. First is money and second is time. That's why we are using bioinformatics. Only two reasons. Okay, data related thing. Okay, model generation, or you can say network study, gene prediction. There are two different things. I'm giving additionally, I'm adding this gene expression also. For example, if you are talking about the genome kind of thing, we have to predict number of promoter region, transcription factor binding sites, exon, intron. It all comes under gene prediction kind of thing. But whether that particular gene is actually expressing or not, this kind of thing comes under gene expression thing. Microarray, you have heard about this thing. So we can write this PSP, protein structure prediction. Either primary structure is not there, sequence is there, secondary structure, tertiary structure, quaternary structure. Have you heard about this term, phylogeny or evolution? This tree kind of structure is nothing but the phylogenetic tree, and we can find out which organism has evolved from which ancestor. This kind of thing we can study by the use of bioinformatics. So I'm listing this thing. Why we are first is the lots of data kind of thing. So there is large amount of data. That's why we are doing bioinformatics. Because it is difficult to handle lots of data in the, for example, lab. For example, I'm taking an example of any student. For example, I'm taking the example of Pratik. Pratik has taken a petri plate in the wet lab experiment, and he has cultured certain kind of colonies into that. After incubation, he has gone somewhere. For example, COVID is there. He has left this particular thing in particular incubator, and he came after, for example, 2 years. That particular culture will survive or not? No, because according to that, there is certain kind of lag phase, log phase, all the things are there. So it will, after a certain period of time, the whole organism or the whole colony will be die. So, but in case of bioinformatics, we can store at any interval of time, at any time. For example, if you are working at 2 p.m., you can do that, or you are working at 4 a.m., you can do that. All the things you can do at any time, and at the same time, you can retrieve the results, which is same as that. As that on the same day which you have performed the experiment. So first thing is large amount of data. In case of bioinformatics, lots of sequencing experiment has been done in 2000. You can say in 1990s to 2010, lots of experiment has been done, and we have generated vast amount of data, and we cannot handle these data by using this wet lab experiment. So to overcome this problem, we need bioinformatics. Second is the cost and time thing. So for setting up certain experiment, we need enzymes or any other proteins which are very costly. But in case of bioinformatics, you need just a system and the resources in the form of internet. So there are various online tools that are available that can reduce your cost as well as time. Third is data redundancy. For example, Shiva has performed this same. For example, someone has performed, some student A has performed this experiment before COVID, and after that Shiva came in the class and he said, "Sir, I want to culture something and I want to grow certain kind of colonies." These are the same colonies which has been developed by this A student, and we do not have to repeat this thing. To overcome this thing, we just have to download our sequence from the database, from the genome, and we can see that most of the thing which we want to identify in that particular genome has been done or not. This kind of thing can reduce the data redundancy. Okay. Next is protein structure and function predictions, in which primary, secondary, tertiary, and quaternary things are there. Majorly, we include these three, that is secondary, tertiary, and quaternary. How can we distinguish primary, secondary, tertiary, and quaternary structure? Primary, secondary, tertiary, quaternary, all is three-dimensional. For example, if you are talking about the primary structure, this alpha carbon is there, this H is there, R is there, and COOH as well as NH2 is there. All the things are existing in the nature in three-dimensional form. Have you heard about this ball and stick model? Everything is three-dimensional. My question is based upon interaction. For example, there are hydrogen bonding, covalent bonding. Based upon that, how can you classify this thing? Primary, secondary, tertiary, and quaternary. So these are the hydrophobic interactions that resulted into folding of protein. Have you seen this kind of structure in case of enzyme? These are active sites. These active sites are nothing but this hydrophobic region that are present due to this, and it resulted into activity of that particular proteins. Have you heard about this thing? All enzymes are protein, but all proteins are not enzymes. So we have to confer whether our structure is resulting into certain functional activity or not. This kind of study can be done by the use of bioinformatics only. So this kind of thing is there. Another thing is evolutionary relationships, graph kind of thing, or you can say tree kind of thing, where this is human, this is, for example, rat is there, this is mouse is there, for example, chimp is there. Actually, chimp should be there or rat should be here. And for example, other monkey is there, for example, any dog is there, anything is there. So this kind of relationships comes under this evolutionary relationship. This is known as phylogenetic analysis, or it is known as cladistic relationship. We will discuss all this thing in our subsequent experiment. I am just informing you why we are doing bioinformatics. Other than that, gene expression thing, and I have also the prediction thing, and I added this thing that is known as expression. So we have to find out whether the original exon which you have confirmed in your genome is actually coding certain kind of proteins or not. So this can be done by the use of gene expression. Next is drug designing. So first thing we have covered about what is bioinformatics. Now we have studied about why bioinformatics, and third question, how can we do bioinformatics? So first, there is lots of data. We cannot analyze that data by using wet lab experiments. So for overcoming that thing, we are using database. There are various databases that are available. I will tell you in the subsequent slides that are there. But based upon database, we have clearly categorized this data based upon either nucleic acid database, or you can say DNA or RNA based database. Second is protein, and third one is metabolic based database. Okay, so this can be done by the use of bioinformatics. Second, by using sequence alignment. For example, I am talking about there are two sequence. There are two different species, for example, S1 and S2, and we have to find out how much they are identical to each other, how much they are similar to each other. This kind of thing can be done by the use of sequence alignment only. Next is known as protein motif, domains, and superfamily. In the sequence thing, for example, if we are talking about sequence, there are certain kind of active regions that are present. These active regions are nothing but the motifs. Okay, these are known as motifs. When we combine motifs, various motifs, then it becomes domains, and when we combine various domains, then it becomes superfamily. So the order is like this: motif, here motifs will, this is sequence, first is sequence, then it is motif, then it is domain, and then it is superfamily. Are you getting this order from primary to quaternary? So these are so in case of bioinformatics, we can easily predict, we can find out various functional regions by the use of motifs, domains, and superfamilies. Other than that, there are phylogenetic analysis we can do for finding out evolutionary relationship. Other than that, microarray is there for finding out gene expression thing. And other than that, for doing drug designing, we can use docking thing and simulation study. So these kind of things we can do by the use of bioinformatics only. So in case of bioinformatics, there are various subdivisions that we have to encounter in our experiments. First is known as sequence similarity. We will perform various experiments regarding BLAST-N, or you can say MSA, multiple sequence alignment in this case. After using this data, we can find out various phylogenetic tree or the ancestral descendant relationship by the use of this phylogenetic analysis. we can predict various genes. In case of, for example, I have given you gene under which how many number of exons are there, how many number of introns are there, how many number of promoters are there, how many numbers of transcription factor binding sites are there. These whole things we can find out by the use of gene prediction only. In case of genome comparison, for example, if we have to compare two different genomes, for example, humans with, for example, rat we have to do, or rat with, for example, mouse we have to do genome wise comparison. Here we are doing gene wise comparison or sequence wise comparison, but here we are using genome. So we have to either compare sequence by sequence or genome by genome. So here we are comparing sequence, and here we are comparing genome. Thank you.